Hello everyone, welcome to the channel. In the previous video, we were studying about male and female reproductive system. And from today's video, we will study the gametogenesis. So gametogenesis is nothing but it is the formation of gametes in the gonads. So in the male gonads, the sperms are formed, male gametes are formed. In the female gonads, female ovum or eggs are formed. Okay, so spermatogenesis is the formation of sperm and oogenesis is the formation of ovum. So that we will study. So first we will start with spermatogenesis. So spermatogenesis is nothing but it is the process of formation of sperms or the spermatozoa in the seminiferous tubules of testis. So now male reproductive system ಕಲಿಯುವಾಗ ನೋಡಿದ್ವಿ testis ಅಲ್ಲಿ seminiferous tubules ಇರ್ತಾವ. So from that seminiferous tubules the sperms are formed. So formation of the spermatids ನಾವೇನಂತ ಕರಿತೀವಿ spermatogonia. That is how the spermatids are formed by the spermatogonia. Sperm mother cell ಇರುತ್ತೆ. So sperm mother cells ಏನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾವ spermatids ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾವ and ಈ spermatids ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ ಆಗಿ sperm ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತಾವ. ಸೊ ಎರಡು ಸ್ಟೇಜ್ ಬರ್ತೈತಿ ಎದ್ರೊಳಗ. ಸೊ spermatogenesis. So spermatogenesis is having two stages: formation of spermatids, that is sperm mother cell will produce spermatids. Okay, so that is spermatogonia will produce spermatids, and this then next stage is spermiogenesis, where spermatids will get transformed into sperm. Okay, so we will study the formation of spermatids and spermiogenesis also. So first, the basic thing that you have to remember is diploid and haploid concept. So during the explanation of life cycle, okay, alternation of generation in plants, that I made different video, there I have explained about this haploid and diploid concept, but again I will repeat. So haploid is nothing but the single copy of chromosome. So where if the gamete is having only a chromosome, so it is not having its pair. Okay, so another copy is not present, then we call it as haploid. So in diploid is nothing but where they have both the copies of chromosome, then we call it as diploid. We call it as 2n. So ಈಗ ಡ್ಯೂರಿಂಗ್ reproduction, in the reproductive cells ಯಾವತ್ತಿದ್ರೂ haploid gametes ಆಗಬೇಕು. ಸೊ ಮೇಲ್ gametes sperm should also be haploid and egg should also be haploid. ಯಾಕೆ ಆ ಥರ ಇರಬೇಕು ಅಂದರೆ now I will tell you. ಈಗ ಬೇಬಿ ಅಂದರೆ ಝೈಗೋಟ್ ಆಗಬೇಕಂದ್ರೆ ಏನಾಗಬೇಕು, ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಸ್ಪರ್ಮ್ ಆ್ಯಂಡ್ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಎಗ್ ಎರಡೂ ಕೂಡ ಏನಾಗಬೇಕು, ಫ್ಯೂಸ್ ಆಗಬೇಕು, ರೈಟ್? ಸೊ ದೆನ್ ವಾಟ್ ವಿಲ್ ರಿಸಲ್ಟ್, ಇಟ್ ವಿಲ್ ರಿಸಲ್ಟ್ ಇನ್ ದಿ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಝೈಗೋಟ್, ಹೌದಲ್ಲ? ಸೊ ಈಗ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಅಂತಂದ್ರೇನು, ಸೊ ಮೇಲ್ ಸ್ಪರ್ಮ್ ಇದು ಏನಿರಬೇಕು, ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಇರಬೇಕು, ಎನ್ ನಂಬರ್ ಇರಬೇಕು. ಅಂದರೆ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟು ಎನ್ ನಂಬರ್ ಇರಬೇಕು ಅಂದಾಗ ಝೈಗೋಟ್ ಏನಾಗ್ತೈತಿ, 2n ಆಗ್ತೈತಿ, ರೈಟ್? ಸೊ ಈಗ ಇನ್ನೊಂದು ಅದು ಎದಕ್ಕೆ 2n ಇರಬೇಕು ಅಂದರೆ ಈಗ ನಾವೆಲ್ಲ ಏನಿದ್ದೀವಿ, ವಿ ಆರ್ ಡಿಪ್ಲಾಯ್ಡ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್, ರೈಟ್? ಸೊ ಡಿಪ್ಲಾಯ್ಡ್ ಇನ್ ದ ಮೀನಿಂಗ್ 2n. ಸೊ ವಿ ಆರ್ ಆಲ್ ಡಿಪ್ಲಾಯ್ಡ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್. 2n ಅಂದ್ರೇನು, 2 times the 23 chromosomes, right? That means we have total 46 chromosomes, ಹೌದಲ್ಲ? ಸೊ 46 ಕ್ರೋಮೋಸೋಮ್ಸ್ ನಮ್ಮ ಕಡೆನೂ ಇರಬೇಕು. ದ್ಯಾಟ್ ಈಸ್ ಎಲ್ಲ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸಲ್ಲೂ 46 ಕ್ರೋಮೋಸೋಮ್ಸೇ ಇರಬೇಕು. ಹಾಗಂದ್ರೆ ನಮ್ಗೆ ನೆಕ್ಸ್ಟ್ ಬೇಬಿ ಫಾರ್ಮೇಷನಲ್ಲೂ ಎಷ್ಟಿರ್ಬೇಕು ಹಂಗಿದ್ರೆ ಕ್ರೋಮೋಸೋಮ್ಸ್ 46 ಕ್ರೋಮೋಸೋಮ್ಸ್ ಇರಬೇಕು. ದ್ಯಾಟ್ ಈಸ್ 46 ಇದ್ದಲ್ಲೇ ನಾವೇನಂತ ಕರಿತೀವಿ, 2 into 23 ಇದೆ ಅಂತ. ಅಂದರೆ 23 ಕ್ರೋಮೋಸೋಮ್ದ ಇನ್ನೊಂದು ಪೇರ್ 2 ಟೈಮ್ ಇರ್ತಾವೆ. ಓಕೆ, ದ್ಯಾಟ್ ಈಸ್ ದ ರೀಸನ್ ಇಟ್ ವಿಲ್ ಫಾರ್ಮ್ 46 ಕ್ರೋಮೋಸೋಮ್ಸ್. ಸೊ ಅದಕ್ಕೆ ನಾವೆಲ್ಲ ಏನು, ಡಿಪ್ಲಾಯ್ಡ್, ಈ ಥರ 2 into 23 ಇರ್ತಾವೆ. ಸೊ ಅದೇ ರೀತಿ ಬೇಬಿಯಲ್ಲೂ ಝೈಗೋಟಲ್ಲೂ 2 into 23 ಕ್ರೋಮೋಸೋಮ್ಸೇ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕು. ಹಾಗಿದ್ರೆ ಅದು ಹೆಂಗೆ ಆಗ್ತೈತಿ, 23 ಕ್ರೋಮೋಸೋಮ್ಸ್ ಮದರಿಂದ 23 chromosomes ಫಾದರ್ ಇಂದ ಕಾಂಟ್ರಿಬ್ಯೂಷನ್ ಆಗ್ತೈತಿ. Okay, that means sperm should get 23 chromosomes and egg should have 23 chromosomes. Then only the 46 chromosomes will be formed in the zygote or the baby. ಅಂದ್ರೆ ಏನು, ಈಗ ಡಿಪ್ಲಾಯ್ಡ್ ಇರ್ತವೆ ನಾವು ಆಲ್ರೆಡಿ, ಫಾದರು ಡಿಪ್ಲಾಯ್ಡ್ ಇರ್ತಾರೆ, 2n ಇರ್ತೈತೆ ಅಂದರೆ 2 times 23 46 ಕ್ರೋಮೋಸೋಮ್ಸ್ ಇರ್ತಾವೆ. ಮದರು 2 times 23 46 ಕ್ರೋಮೋಸೋಮ್ ಅಂದರೆ ಇವರು ಡಿಪ್ಲಾಯ್ಡ್, ಮದರು ಡಿಪ್ಲಾಯ್ಡ್. ಸೊ ದ್ಯಾಟ್ ಈಸ್ ದ ರೀಸನ್ ದೇ ಹ್ಯಾವ್ ಟು ಬಿಕಮ್ ಹೆಪ್ಲಾಯ್ಡ್. ಅಂದರೆ ಓನ್ಲಿ ಒನ್ ಪೇರ್ ಆಗಬೇಕಂದ್ರೆ ಅವ್ರೇನು ಮಾಡ್ತಾರೆ, ದೇ ಶುಡ್ ಅಂಡರ್ಗೋ ಮಿಯೋಸಿಸ್. ಓಕೆ, ಸೊ ಮೈಟೋಸಿಸ್ಸಿಂದ ಏನಾಗ್ತೈತಿ ಅಂದರೆ 2n ಇದ್ದಿದ್ದು 2n ಆಗೇನೆ ಉಳಿತೈತಿ, ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ ಡಿಡಕ್ಷನ್ ಆಗಲ್ಲ. ಬಟ್ ಮಿಯೋಸಿಸ್ಸಲ್ಲಿ 2n ಇದ್ದಿದ್ದು, ಸೊ ಮೈಟೋಸಿಸ್ಸಲ್ಲಿ 2n ಇದ್ದಂಥ 2 ಸೆಲ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾವೆ. ವೆರಾಸ್ ಮಿಯೋಸಿಸ್ at the final stage 2n ಇದ್ದಿದ್ದೇನಾಗ್ತೈತಿ n ಅಂತೇಳಿ, ಸೊ 4 n ಇರುವಂಥ ಹೆಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತವೆ. ಸೊ ಮೈಟೋಸಿಸ್ ಆ್ಯಂಡ್ ಮಿಯೋಸಿಸ್ ಇಟ್ ಈಸ್ ದ ಡಿಫ್ರೆನ್ಸ್. ಓಕೆ, ಸೊ ಹಾಗಾಗಿ both the egg as well as sperm, they should have n and n, that is haploid number of chromosomes. 2 times n ಇದ್ರೆ ಏನಾಗ್ತೈತಿ, 46 ಇತ್ತಂದ್ರೆ ಸೊ ಬೇಬಿ 46 ಆಗಲ್ಲ, ಸೊ ಝೈಗೋಟ್ 46 ಆಗಲ್ಲ, ಅದು ಅಬ್ನಾರ್ಮಲ್ ಆಗಿಬಿಡತ್ತೆ. ಸೊ 46 ಡಿಪ್ಲಾಯ್ಡ್ ಅದು ಆಗಬೇಕಂದ್ರೆ ಇವರು 23. ಸೊ ಮೇಲ್ ಶುಡ್ ಆಲ್ಸೋ ಕಾಂಟ್ರಿಬ್ಯೂಟ್ 23, ಫಿಮೇಲ್ ಶುಡ್ ಆಲ್ಸೋ ಕಾಂಟ್ರಿಬ್ಯೂಟ್ 23 ಟು ಫಾರ್ಮ್ 46 ಇನ್ ದಿ zygote. ಅದಕ್ಕೆ they have to be haploid in nature. So ಅದಕ್ಕೆ you can see here. So the spermatogonia in 2n ಇದೆಯಲ್ಲ ನಂಬರ್, ಸೊ it is the mother cell. ಸೊ ಅದಿನ್ನು ಡಿವಿಷನ್ ಆಗಲ್ಲ. ಸೊ it is the spermatogonia, sperm mother cell ಅಂದ್ಕೊಳ್ಳಿ, ಫಿಮೇಲ್ ಅಂತ ಅಂದ್ಕೊಳ್ಬೇಡಿ. ಸೊ it is sperm mother cell. Okay, so when sperm mother cell which is 2n, that means it is diploid. Okay, so obviously we all are having diploid cells, right? Because we all have 46 chromosomes. But now during reproduction, since the zygote has to form, so that means male should contribute n number of chromosomes and female should contribute n, then only 2n will form. Okay, then only baby will also be 2n and like us, right? So that is the reason when 2n spermatogonia, it will first undergo mitosis. As I said, mitosis will not reduce in the chromosome number, so 2n primary spermatocytes will only form, but then it will undergo meiotic 1 forming n secondary spermatocytes, and then it will undergo meiotic 2 which will form spermatids, and then spermatogenesis will occur to form spermatozoa. Here you can see. So first the sperm mother cell, it will undergo what? Mitosis. So it is undergoing mitosis to produce 2n only, because mitosis will not reduce the number of chromosome. 2n will, so one 2n cell will produce 2n cell. Okay, but then this primary spermatocyte, it will undergo meiosis 1. So what it will undergo? Meiosis. So this meiosis will help in producing the haploid n cells, right? What they will produce? Haploid n cells, and these haploid n cells will undergo meiosis to produce the spermatids, right? So these spermatids will undergo spermatogenesis and they transform into the sperms. So like this, so that means what? One primary spermatocyte will able to produce 4 spermatids or spermatozoa. Okay, so from one primary spermatocytes 4 sperms can be produced. So this is how while studying the genetics concept in detail, we will study about mitosis and meiosis, and you just have to know that how the chromosomal, yeah, what do you mean by N, what do you mean by diploid, and what do you mean by haploid. So spermatids, which is 2n in number, it will undergo mitosis to produce primary spermatocytes, and meiosis 1 will take place to produce secondary spermatocyte, which will be in n in number. Okay, and then each n will undergo again meiosis to produce spermatids, which will undergo spermiogenesis to produce spermatozoa. Okay, so a spermatocyte, a primary spermatocyte will produce secondary spermatocyte, and a secondary spermatocytes will produce spermatids, 2 spermatids. Thus one primary spermatocyte will produce 4 spermatids or sperms. So how they are produced, here you can see. So a portion of seminiferous tubules. Okay, so here there is spermatogonium, right, sperm mother cell, right, and primary spermatocyte it is produced, and then from that secondary spermatocytes are produced. Okay, so there is a spermiogenesis taking place, so meiosis is taking place here, and then spermatids are produced. So the spermiation, after spermiogenesis, sperm heads are embedded in the Sertoli cells to get nourishment. So they are released to the lumen of seminiferous tubules. It is called as spermiation. So after this spermiogenesis, when the sperms are produced, okay, so then the sperm heads, they are embedded in the Sertoli cells. As we have studied, Sertoli cells will help in the nourishment. So Sertoli cells will be there, and the sperm heads, they are dipped in the Sertoli cells for the nourishment. Okay, so that you have to know. So they are released into the lumen of seminiferous tubules. This we call it as spermiation. Okay, so next, spermatogenesis. We have to see here, the hypothalamus secretes the gonadotrophin releasing hormone, which acts on the pituitary. So which secretes 2 gonadotrophins, which is luteinizing hormone and follicle stimulating hormone. So luteinizing hormone, it acts on Leydig cells, which secretes androgen, and it stimulates the spermatogenesis. And the follicle stimulating hormone, which acts on Sertoli cells, and it stimulates the secretion of factors of spermiogenesis, which are required for the spermiogenesis, okay, for transformation of the spermatids into the sperm. Okay, so this is the role of hormones in the spermatogenesis. So how the hormones will contribute? So first, the hypothalamus, it secretes the gonadotrophin releasing hormone, so that will act on the pituitary, and that will secrete 2 gonadotrophins. Okay, so and that, yeah, which are they? One is luteinizing hormone and another one is follicle stimulating hormone, and then LH, luteinizing hormone, which will act on Leydig cells, and they will help in secreting the androgen, so which stimulates androgen. Why it is required? For stimulation of spermatogenesis. And then follicle stimulating hormone, this will act on Sertoli cell, and it stimulates the secretion of factors which are needed for the spermiogenesis. So this slide is also very important to understand the role of hormones in the spermatogenesis. Okay, so spermatogenesis, now you can see the structure of sperm. So it is having acrosome, which is head piece, which is middle piece is mitochondria, and principal piece and the end piece. So the length also is sometimes important, and mature sperm is about 60 micron or 0.06 millimeter in length. Sperm is enveloped by the plasma membrane, and the sperm has 3 regions: head, middle piece and the tail. So head, first one is head. So oval shaped, formed of nucleus and acrosome. So it is formed of nucleus and acrosome. So here it will be, you can see the nucleus. Okay, so acrosome, and acrosome is formed of Golgi complex. It contains lytic enzymes. So when the sperm has to enter, then it requires some lytic enzymes. Okay, so when sperms enter the egg, okay, so it that is why there is acrosome. It is in the front, and it is formed from Golgi complex. So there is a Golgi complex there which contains the lytic enzyme. So behind the head is, which is neck. Okay, so neck it is there, and then it is a very short part. Okay, and then middle piece, it is composed of axial filament which is surrounded by mitochondria and cytoplasm. So mitochondria, it produce energy for the sperm motility. As we know, sperm should move, right? So that is the reason, to produce energy, the mitochondria is present, which is present in the middle piece. Okay, so mitochondria, axial filament which is surrounded by mitochondria and cytoplasm. Okay, so for the energy for the sperm motility. Next comes the tail, and it consists of central axial filament. Okay, the sperm moves in the fluid medium and female genital tract by the undulating movement of the tail. Okay, so the parts and the sperm diagram is also very important, and the length also you should know, and what is the importance of the regions, that also you should know. So mitochondria energy produce ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತೈತಿ, and head acrosome ಇರುತ್ತೆ. acrosome ಅಲ್ಲಿ Golgi complex ಅಂತ ಇರ್ತೈತಿ. So that Golgi complex will help in ಲೈಟಿಕ್ ಎನ್ಸೈಮ್ಸ್ ಅಂತ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ ಫಾರ್ ಸ್ಪರ್ಮ್ ಎಗ್ಗನ್ನು ಎಂಟ್ರಿ ಆದಮೇಲೆ, ಫೀಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸಿಸ್ಟಮನ್ನು ಎಂಟ್ರಿ ಆದಮೇಲೆ, ಸೊ ಇಟ್ ರಿಕ್ವೈರ್ಸ್ ಲೈಟಿಕ್ ಎನ್ಸೈಮ್ಸ್. ಓಕೆ, ಸೊ ಟು ರೀಚ್ ದಿ ಎಗ್. ಓಕೆ, ಸೊ ಅದಕ್ಕೆ ಎಗ್ಗನ್ನು ಅದು it contains lytic enzymes. Okay, so this is very important. Okay, so in human, so now sperm, other structure and what are the parts that we have studied, and now we will study the human male ejaculate. So ಎಷ್ಟು ejaculation ಆಗ್ತೈತಿ, ಎಷ್ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತೈತಿ. The human male ejaculates about 200 to 300 million sperms during the coitus. Okay, so that means when there will be sexual intercourse, so it produces about 200 to 300 million sperms. And for normal fertility, at least 60% of the sperms must have normal shape and size, and 40% must show vigorous motility. Okay, so at least 60% of the sperm should have normal shape, size, movement, etc. Okay, so this is about spermatogenesis. I hope you understood the topic. So the role of hormones and how the hormones are produced. Okay, so this everything is important. So that's it for today. Thank you so much. Watch the next video for the oogenesis. So please hit the like button if you really like the video. Share it to the one who is in need, and subscribe to the channel if you have not subscribed. Thank you so much.